Okay, DNA replication. This is the process by which a new DNA are generated. Okay, so replication. This is a biochemical process by which a DNA produce the exact duplicates of themselves. So they are in this process of replication, the parent DNA will be producing two daughter DNA. Okay, so mapuproduce siya ng dalawa. And we had summarized the steps in DNA replications. First, the enzyme DNA helicase causes the two strands of DNA to unwind, which produces two templates strand. The point at which the DNA double helix is unwinding is called the replication fork. Okay? And then we also have free nucleotides pair with the complementary base on the template strand by means of hydrogen bonding. Okay? Base pairing involves cytosine pairing with guanine pa rin. Okay, so the C will pair with G, and then the adenine will be pairing with thymine. Okay? Pairing occurs one nucleotide at a time. Next, the DNA polymerase joins the newly attached nucleotides to create a continuous strand, or this is known as the leading strand, in the 5' to 3' direction. Also, aside from joining this new nucleotide together, DNA polymerase also verifies if the base pairing is correct. So, hindi lang niya ina-attach together, rather, tinitignan pa rin niya kung correct ang base pairing that the cytosine will pair with guanine and adenine will pair with thymine and vice versa. Okay, so, check din niya before it catalyzes the formation of phosphodiester linkage between these nucleotides. And also, other strand is formed in short segments, or these segments are called as Okazaki fragments or the lagging strand in the 3' to 5' direction. Kasi nga, the DNA polymerase could only act upon 5' to 3', so yung nasa 3' to 5' ay segmented. Okay, segmented siya. However, meron tayo, dahil sa tulong ng DNA ligase, these segments can be joined together such that the gaps known as the nicks will be bonded together by this DNA ligase. Okay, so let's visualize it. So these are the steps. So we have step 1. So the step 1, we all know that the DNA is composed of a double helix structure, and the nucleotides here are bonded by a hydrogen bond. Nag-base pair siya, rather, it has base pairing because of the attraction hydrogen bond between these nucleotides. Kaya siya meron ding coiled part. And for the action of DNA helicase, this DNA causes to unwind. So, mag-unwind siya. Maghihiwalay sila. Okay? So, this is the old strand, 5' to 3'. Okay? It's the old strand. And we also have its complementary strand. This is the 3' to 5'. Okay? So, as you could see, the unwinding starts at the replication fork. Ito daw yung replication fork niya. Okay? Upon this unwinding, makikita yung mga nucleotides. Okay? So, the A is a complement with T. The T is complement with A. That's the G. Ito yung complementary base pair niya na C, T, and A. Okay? So, with this exposed nucleotide in this template strand, so ang tawag natin dito sa old old DNA na to ay template or this is the pattern strand. Okay, so that is step number 1. The helicase, DNA helicase, causes the DNA strands to unwind, okay, producing 2 template strands. So, naka-expose na yung mga nucleotides nila dyan. Okay, and this is now the replication fork. For step number 2, there are free nucleotides around inside this nucleus. So may mga free nucleotides. Ayan, may mga free nucleotides siya. And upon unwinding of this DNA, these free nucleotides will have a base pairing with the nucleotides inside the template strand, DNA template strand. So, if there's the nucleotides na naka-expose dyan sa template strand, yung free nucleotides will base pair with the nucleotides in the template strand. So, with this base pairing, of course, hydrogen bond is responsible for this. Pero ang base pairing would come one at a time. So, ito, the A will base pair with T, the G will base pair with the free nucleotide C. Ganon din dito sa other strand. So, the T will base pair with A, the G will base pair with C. And this happens one at a time. For step 3, okay, that's step 3, the DNA polymerase will now act upon on this base paired nucleotides. So, it will now join the newly formed the DNA polymerase will now act upon on these free nucleotides being base paired with those on that template to form a leading strand. So, pagkokonektahin na niya itong mga free nucleotides na newly formed or newly base paired with the template or the DNA pattern so that it will now be producing, so magkakaroon na once the DNA polymerase bond these nucleotides together, it will now be forming a new continuous strand. So this is known as the leading strand. It acts in the 5' to 3' direction. Okay. Aside from joining these nucleotides newly attached, newly bonded to the template strand, okay, chine-check din ni DNA polymerase kung tama ba yung pagkaka-base paired nito. So this is GC, that is still GC, and that is TA. Okay? So ulitin natin. For step number 2, for step number 3, since there are free nucleotides being attached or being bonded with a hydrogen bond to the nucleotides in the old strand, okay, these nucleotides are now attached or are now connected via phosphodiester linkage so that it will be producing a long strand known as the leading or the continuous strand. Okay. Ganun din dun sa other strand here. The DNA polymerase will attach together the free nucleotides being paired with the old DNA strand. However, since this is in a 3' to 5' direction, hindi siya nakaka-produce ng continuous. Okay? So, for this case, okay, pili lang tayo ng ito. Okay, so for this case, si DNA polymerase still checks if the base pairing is correct, still joins together this newly attached nucleotide. However, since this is in a 3' to 5' direction, hindi niya completely na a-attach or together itong mga nucleotides such that nagkakaroon ng mga gaps. Hindi siya continuous. So this is a lagging strand or Okazaki fragment. So fragmented yung maproproduce ng DNA polymerase dito sa 3' to 5' direction or new strand. So, for step 4, we will be needing the DNA ligase to act upon on this fragmented strand. Kasi nga hindi dapat na fragmented or non-continuous yung strand. So, with the help of DNA ligase, these gaps will be closed. These gaps is known as the nick. Okay, so the DNA ligase will act upon on these gaps, on these fragmented or Okazaki fragments, so that in the end of the replication, this still will be producing a continuous strand. Okay, so as a summary, after the replication, that this old DNA is now producing two daughter DNA or two composed of, so ang ating product dito ay two DNA strand with identical base pairing. Okay, so tingnan natin ito. This is the TA and it will have a T. This is the GC. Okay, balikan natin dito sa umpisa. Okay, so this is TA. And of course, this is ATA then. TA, TA, GC, GC. So this is GC, hindi lang naipakita. This is TA, GC, and then this is also GC. So as you could see, the product after the DNA replication are identical DNA. Kasi nga, yung strand nila ay even though yung template strand nila ay magkaiba, because of the base pairing, magkakaroon pa rin tayo ng identical DNA. So whatever is the property of the old DNA, ganun din yung maproproduce na daughter DNA.